Imagine that you are traveling to Japan, but you only speak English. Someone hands you a map which is written entirely in Japan. You have the instructions, but you can't understand them. You need a translator in such situations. Inside every living cell, a similar problem exists. DNA and the mRNA speak the language of nucleotides, which is written in AUGC or ATGC, whereas the proteins are built using amino acids. These two are completely different biological languages. So to basically translate one language to the other, we need a translator. So that translator is the transfer RNA or tRNA. Transfer RNA or tRNA is a small RNA molecule that acts like the cell's molecular translator. So why do you need a translator? As I told before that the genetic code is written in 3-letter codons, whereas that's the mRNA, and the proteins are written in amino acids. So the conversion, right, to convert the language of 3-letter codons, which is there on mRNA, to the amino acids, you require a translator. That translator is transfer RNA. And why you call it as transfer RNA is because it literally transfers, or it carries something from one place to another. tRNA actually picks up those specific amino acids and then transfers it to the ribosome where proteins are being built. So without having tRNA, the genetic code will just remain in the DNA. It would not become a functional protein. Alright. So let's dive in further and see why tRNA has to be incredibly accurate. So to be a real translator in the world, you should actually understand two different languages. Similarly, tRNA understands two biological languages. One, which is the mRNA codons, the language of nucleotides. The other end, it carries specific amino acid, which is the language of proteins. So it basically, the tRNA connects these two languages together. Okay, it helps in translating. It translates the genetic information into proteins. But tRNA is like the intelligent messenger. It understands the both languages, okay, and it brings the right amino acids at the right time. That is why it is called as the molecular translator. So what does tRNA actually look like? So the tRNA is made of around, it's a very small RNA molecule. It's made about 70 to 90 nucleotides. The most common shape is this 2D clover leaf when it's drawn on paper. But actually on the cell, it looks like a compact L-shaped three-dimensional structure. So the structure basically allows the tRNA, this unique structure which you see, right? This allows tRNA to perform its function, two important tasks. So the first important task is that at one end there is the anticodon loop or the anticodon arm, which basically recognizes and pairs the specific mRNA codon. At the opposite end, we have the acceptor stem, which basically carries the correct amino acid. So in simple terms, one end reads the genetic instructions, whereas the other end delivers the matching amino acid to the ribosome. Now let's see actually how does tRNA does its job. What is its job actually, right? So basically the first job is charging or loading. So before translation or before you synthesize proteins, there's a special enzyme called aminoacyl tRNA synthetase, which basically uses ATP, and its job is basically to attach the amino acids to the attach the correct amino acids to the tRNA. Okay. So this creates a charged tRNA which is ready for delivering its cargo. Step 2 is the codon recognition. So the charged tRNAs which are there, once it enters the ribosome, the anticodon will then pair up with the mRNA codon which is there on the mRNA, and this will ensure that the correct amino acid is selected according to the genetic code. And the third step is that ribosome will form a peptide bond, adds the amino acids, okay, to the growing chain, protein chain. So then the empty tRNA is then, it will exit the ribosome. It will again come reload with another amino acid and the cycle repeats. So the basic three important roles or tasks of tRNA is to first, it decodes the genetic message. It reads the codons on the mRNA using its anticodon. And second, it acts like a courier. It basically picks up the correct amino acid and safely carries it to the amino acid, carries it to the ribosome. Finally, it will deliver the cargo. It will place the amino acid in exactly the correct position so that it can be added to the growing protein chain. Right? Now let's understand that the transfer RNA isn't just for making proteins. It's also opening up new exciting possibilities in medicine and biotechnology. So scientists are exploring engineered tRNAs to help treat genetic diseases caused due to premature stop codons. Okay, they allow actually produce complete functional proteins. Second, researchers are designing tRNAs which can correct certain genetic mistakes. And then in synthetic biology, modified tRNAs are helping to create proteins with entirely new properties for medicine and biotechnology. And tiny fragments of tRNA is designed as biomarkers for detecting diseases like cancer and neurological disorders. So tRNA is much more than a translator, right? So let's recap everything together. Transfer RNA or tRNA is the cell's molecular translator which connects the language of nucleotides with the language of proteins. Its unique structure allows it to do its function, okay, but it allows it to recognize the mRNA codons and also carry the correct amino acid. By reading the genetic code, transporting amino acids, and delivering them to the ribosome, tRNA ensures that every protein is built accurately. But remember that tRNA is not just doesn't work alone. It does it along with a remarkable molecular machine that reads the genetic message and joins the amino acids together and makes the protein. So that's the job done by ribosomes. So in the next video, we will be covering and exploring about ribosomes. So if you have really liked the video, please don't forget to like, share and subscribe to the channel. Thanks for watching. I'll see you guys in the next lecture. Thank you.